which is needed in anaerobic oxidation. And the second one is the lipid metabolism. The third one is the mitochondrial oxidative phosphorylation reactions. We have many types, many other types like myoadenylate, deaminase, or creatine phosphate. These are another sources to take energy for exercising. But the first, most important one is anaerobic oxidations. Anaerobic oxidation. دكتورة حنان وصلت؟
ايوه السلام عليكم دكتور علي بيه.
سلامة الله. اهلا دكتورة حنان اتفضلي.
السلام عليكم ازي حضرتك؟ ازي حضرتك؟
سلامة الله اهلا مساء الخير يا فندم. Now we will present our eminent Professor Hanan, Professor and Head of Rheumatology, Medicine, Rheumatology and Immunology in Bani Suef University to talk about metabolic myopathy, and after that I will give some key points about the subject. Please, Dr. Hanan, you have to start.
Thank you. Can I share my screen, دكتور شيماء؟
ماشي اوكي اتفضلي.
Okay.
فيصل زيد عيط على البنات.
My screen is shared now.
Yeah.
Okay.
Yes,
السلام عليكم ورحمة الله وبركاته. Thanks Professor Ali and his team for giving me this chance. Now our topic is about metabolic myopathies. Myopathies include many forms of myopathies: inflammatory myopathies, dystrophic myopathies, endocrine, infectious, parasitic, drug-induced, neuromuscular disease, and metabolic myopathies. Metabolic myopathies, muscle disease, result from inborn errors of metabolism, either in carbohydrate metabolism, lipid metabolism, or mitochondria. Carbohydrate metabolism, either glycogen synthesis or breakdown, and glycolysis. Lipid metabolism, fatty acid oxidation, mitochondria, Krebs cycle or electron transport chain. This slide is very important. Nearly if we illustrates this, we can understand what's going on forward. This is the glycogen, this is glucose. Glycogen is broken down into glucose 1-phosphate. The defect in the glycogen storage disease lies in these areas. Either in the glycogen synthesis from glucose, the defect in the branching enzyme, like glycogen synthase enzyme, here the defect in the glycogen synthesis, or the defect in the glycogen breakdown, or the formation of glucose from glycogen, like the debranching enzyme defects, or in the glycolytic pathway, or the formation of pyruvate from glucose. So the glycogen storage disease lies in 3 areas, either in the glycogen synthesis, glycogen breakdown, or glycolytic pathway. The end stage of formation of glucose is the formation of pyruvate from glucose to enter the mitochondria to form energy. Pyruvate also comes from lipids. When the glucose finishes, the body recalls for triglycerides, fatty acids, and pyruvate to enter also the mitochondria. So pyruvate comes from 2 sources, either from glucose or from free fatty acids. Now, pyruvate enters into the mitochondria in conjunction with what is called the carnitine shuttle. So acetyl coenzyme A doesn't enter the mitochondria by itself. No, it enters in association with carnitine through carnitine shuttle. It undergoes 3 stages. First stage is beta oxidation of fatty acid. Second stage is Krebs cycle or citric acid cycle. And third stage is the respiratory chain or the formation of adenosine triphosphate or the energy. So these are the whole metabolic process starting from glycogen or fat, ending in formation of energy. Here is glycogen storage disease. Any defect in the lipid is the lipid storage disease. Any defect in the mitochondria is the mitochondrial myopathies. These are illustration for the phases of beta oxidation of fatty acids, 3 stages: beta oxidation of fatty acids, then respiratory chain or electron transfer chain inside the mitochondria. This is the carnitine shuttle I said before. Acetyl coenzyme, acetyl CoA in the previous slide cannot enter the mitochondria. These are the mitochondrial membrane, the outer mitochondrial membrane, the inner mitochondrial membrane. This carnitine shuttle can allow acetyl coenzyme A to enter into the mitochondria to undergo the 3 stages of beta oxidation, then Krebs cycle, then formation of the respiratory chain or the ATP. For the first 20 minutes of exercise, the body typically relies on glucose derived to the cell and generated from glycolysis to generate acetyl-CoA that feeds the citric acid cycle. If activity continues, the concentration of glucose diminishes and the body transitions to breakdown of triglycerides from ATP for beta oxidation of fatty acids. In cases of maximal exertion, the energy demands exceeds the oxygen supply. So some of the pyruvate is reduced to lactate to allow for anaerobic glycolysis. When lactic acid accumulates, lactic acidosis, pain, and fatigue occurs. So we start with the glycogen metabolism, disorders in the glycogen metabolism. These are inherited disorders. The age of onset varies from birth to adulthood. Muscle symptoms induced by either brief exercise, such as lifting heavy weights, or less intense, sustained dynamic exercise, such as swimming, climbing stairs, or running. Eventually, acute muscle breakdown may lead to myoglobinuriae, cramps, and muscle swelling. This is called metabolic crisis. Glycogen storage disease result from deficiency of any of 3 pathways. As we said, errors in glycolysis or the debranching enzyme, errors in the glycogen synthesis or the glycolytic pathway. Here, or here. Glycogen storage disease are called GSD and enumerated in Roman numerals from 1 to 12. These are the 12 GSD or glycogen storage disease from 1 to 12. This is another simplified collection of glycogen storage disease, the famous one. The famous ones are named according to the physician who described them. Type 1 or von Jerkes disease, the problem is in the glucose 6-phosphatase. Glucose 6-phosphatase is one of the glycolytic pathway. Here in the glycolytic pathway, in the middle, the patient in any glycogen storage disease suffers in 2 areas. On the area of carbohydrate metabolism, he suffers from either hypoglycemia or even hyperglycemia, and the other area in the exercise. So we have 2 problems in glucose and in exercise tolerance. In glucose 6-phosphatase or von Jerkes disease, the patient has severe hypoglycemia because he cannot transform glucose into pyruvate. Severe hypoglycemia. He has lactic acidosis because he cannot enter the respiratory chain, so pyruvate is transformed to lactic acid. Hepatomegaly, hyperlipidemia, hyperuricemia, short stature, doll-like faces, protuberant abdomen, but the glycogen structure is normal because the defect is in the glycolytic pathway. Pompe's disease or type 2 is a lysosomal alpha-1,4 glucosidase. This enzyme acts on glycogen-like structures inside the lysosome. So it leads to accumulation of glycogen-like material in inclusion bodies in the lysosomes, in the heart, in the muscle, cardiomegaly, leading to muscle weakness. Curie's disease is the defect is in the glycogen debranching enzyme. Here, the debranching enzyme. So there is hypoglycemia and liver enlargement. He has the glycogen is abnormal. Anderson disease, the problem is in the branching enzyme. Here in this area is in the branching enzyme.
من فضلك يا دكتورة حنان
The child suffers from liver cirrhosis. Yes.
تمام. في ناس في الويتنج روم بس او كده. ماشي اوكي. شكرا.
What can I do? I allow for them.
كمان لا اتفضلي خلاص تمام. الدكتوره شيماء
كمل.
شكرا اتفضلي. شكرا معلش.
شيماء can manage. Okay. Here the Anderson disease, the problem is in the branching enzyme. Now we come for the McArdle disease, or the problem is in the glycogen phosphorylase. Here, the glycogen phosphorylase. Phosphorylase enzyme, one in the liver and one in the muscle. In McArdle disease, the problem is in the muscle glycogen phosphorylase, or the glycogen degradation pathway. So he suffers from muscle cramps and hypoglycemia. The glycogen structure is normal. Here's the disease, or type 6. This is not a myopathy because the defect is in the hepatic glycogen phosphorylase. The problem in the arm of the branching enzyme defect or the glycogen synthesis, the patient suffers hypoglycemia, not hypoglycemia, but not like diabetes because glucose can enter the cell. There is no defect in insulin. So these patients has transient hyperglycemia. However, if the defect in the debranching enzyme, like the glycogen phosphorylase, the problem is hypoglycemia because the body cannot get glucose from degradation of glycogen. This is a brief idea. This is some details about the von Jurgens disease. The defect in von Jurgens disease in the glycolytic pathway, as we said, in the glucose 6-phosphatase enzyme. The glycogen deposits in the liver. There is hyperuricemia, hyperlipidemia, fatty liver, and so on. Pompe's disease, like we said, it is a lysosomal 1-alpha glucosidase. The deficient enzyme normally resides in lysosome and is responsible for digesting glycogen-like material in endosomes. It is more similar to disease like Tay-Sachs disease. The tissues most severely affected that normally have glycogen stores. This occurs before 2 years of age. McArdle disease, which is relatively common, the defect is in the myophosphorylase inside the muscle. Here is an example of a 25-year-old woman had a lifelong history of exercise intolerance that was often accompanied by episodes of cramping. The episodes were somewhat ameliorated by drinking sucrose-like soft drinks immediately before exercise. The latest episode occurred during her first spin class, stationary bicycling with resistant food at her local bicycle shop. She initially had extreme weakness in both legs and muscle cramps, and later secreted red-brown urine. So she entered into rhabdomyolysis. In subsequent sessions, in addition to the high sucrose drink, she reduced the load on the bicycle and was better able to tolerate the initial phase of exercise. After 10 to 15 minutes, she experienced a second wind and was able to continue her exercise successfully. What about the story of the second wind? Initially, the body can use glucose to form the energy. After some time, glucose is insufficient here because the patient has a problem of glycogen degradation or the glycogen phosphorylase enzyme in McArdle disease. So the body restores to formation of energy from fat. So muscle pain is relieved. This is called the second wind. The patient improves after some time in glycogen storage disease. These patients suffer from hypoglycemia. They improve after some time due to formation of energy from fat. This is what is called second wind. This is characteristic of glycogen storage disease, whichever the site of defect. This is the initial diagnostic test of any metabolic myopathies. This is the forearm exercise test, 1 minute hand clenching, blood samples collected pre and post exercise, attenuated blood lactate initially, which increases post exercise. Why lactate increase at the end of the exercise? Because there is no aerobic glycolysis, it is blocked. So anaerobic glycolysis occurs, leading to increased blood lactate. Decreased pyruvate, lower glycolysis rate, normal increase in ammonia production. It is highly sensitive, highly specific to rule glycogen storage disease, to rule in or out. This is be called a screening test. if you suspect from the history a glycogen storage disease. When you suspect the patient had hypoglycemia, has pain, muscle weakness initially at the first minutes of exercise. Initially, this characterizes glycogen storage disease. Now we come to the lipid disorder. The lipid disorders comes in second. The lipid metabolism recalled after we consume glucose initially in exercise. So the patient suffers from pain, no second wind. The pain is prolonged. The pain is after intense exercise. Not like glycogen storage disease. This figure illustrates how the acetyl coenzyme A enters the mitochondria. This is the mitochondria, the outer mitochondrial membrane, the inner membrane. This is the carnitine shuttle with acetyl coenzyme A, and then enters into the Krebs cycle and the respiratory chain cycle to form ATP. This is the site of genetic defect. This is a comprehensive illustration, site of genetic defect in lipid storage disorders. These are the names of the lipid disorders, either in the carnitine cycle disorder, defect in the beta oxidation enzyme, or neutral lipid storage disease and lipine-1 deficiency and deaminase deficiency. These are the sites of deficiencies. These are the fatty acid oxidation disorders. According to the enzyme defect, very long chain, carnitine transporter deficiency. This is the gene defect. This is the prevalence. These are the symptoms named after the site of organ. L for liver function, G for glycemia, for hypoglycemia, M for skeletal myopathy, R for rhabdomyolysis, C for cardiomyopathy. And these are the complications. Here, how can we diagnose? We can diagnose by the pattern. The pain comes later in exercise. This is the first. Second, during the acetyl coenzyme A is elevated because it cannot enter the cell. Defect in protein responsible for transport oxidation fatty acids to generate acetyl-CoA. Fundamental issue is the utilization of fat as the energy source. The presentation is acute. Symptoms can be similar to that seen with glycogen storage disease, but pain and cramping with prolonged fasting and exercise. Here the patient is affected by prolonged fasting also. Rhabdomyolysis may occur following acute attack. Symptoms appear in conditions of glucose or glycogen depletion. Serum glucose, serum lactate, CK normal between each acute attack. Sometimes there may be associated manifestations like cardiomyopathy or hepatic dysfunction. Here again is the mitochondrial membrane, the outer, the inner mitochondrial membrane. These are the diagnostic test is acyl carnitine profile test. What is that? The deficiency of beta oxidation lead to build up of acyl carnitine species. The acyl carnitine is elevated. Another more complex test only performed if fatty acid oxidation defect highly suspected. You cannot perform this test except if you suspect that this myopathy is due to fatty acid oxidation defect. Like in the history, the patient suffers after prolonged fasting, after intense exercise. It's most sensitive in fasted state during the acute attack. Then you can come to the genetic test for a specific variant indicated with positive acyl carnitine profile. Then positive acyl carnitine profile is elevated acyl carnitine relative to carnitine level. The treatment here is low fat, high carbohydrate diet. Avoid prolonged periods of fasting. Carbohydrate loading may be beneficial prior to prolonged exercise and also supplemental prolonged exercise with regular carbohydrate intake. All treatment of metabolic myopathies take care of two aspects. The exercise adaptation, I should learn the patient how to exercise, and the diet, whether the diet low in fat or high in carbohydrate and so on. These are examples of fatty acid oxidation myopathies, CPT2 deficiency. This is another type, very long chain acyl dehydrogenase deficiency, deficiencies of different sites leading to this defect. And lastly, we come to the mitochondrial myopathies. The mitochondrial myopathy is the most deleterious form because the mitochondria is the factory, is the site of respiratory chain, is the organelle which forms oxygen. So it affects many organs, not only the muscles. It affects the heart, it affects the CNS, it affects many organs. Collective term to describe defects in enzymes in mitochondrial pathways. The inheritance pattern, either maternal inheritance or autosomal recessive. It is a diverse group of genetic disorders with variable clinical presentation depending on the enzymatic defect. It means not necessarily the patient has full-blown picture. He may be heterogeneous. Mitochondrial myopathies present with a wide range of clinical expression. Organ system relying mostly on aerobic metabolism are affected. The nervous system is common. When skeletal muscle is affected, either alone or with central nervous system disease, the term mitochondrial myopathy is used. The clinical features many complex names, initials, either isolated myopathy or progressive external ophthalmoplegia or CAO, Leber hereditary optic neuropathy, severe encephalomyopathy, MELAS, MERF, MIDD, Minji, NARP, Pearson syndrome, coenzyme Q10 deficiencies. Now, the evaluation of metabolic myopathies, we have brain imaging, exercise testing, EEG, lastly, muscle biopsy, genetic studies, lab studies. This is the exercise test. Again, in mitochondrial myopathy is different from the exercise test in glycogen storage disease. Here we do a progressive exercise test. We can find lower oxygen. The oxygen cannot rise at the end of the test. Poor specificity, high false positive rate. Forearm exercise test with venous blood gas measurement. You cannot, you usually don't order for blood gases except if you suspect mitochondrial disorder. So you withdraw oxygen and CO2 at the start of exercise and after that. With exercise, oxygen concentration should fall due to increased oxygen consumption. However, with mitochondrial disorder, oxygen concentration remains close to the resting condition. Oxygen gas does not change at the start of the test and at the end of the test. This is characteristic of mitochondrial myopathy as regard to exercise testing. This is the muscle biopsy, which is characteristic for mitochondrial myopathy. Ragged muscle fibers. Here, the mitochondria at the periphery of the muscle fiber due to accumulation of abnormal mitochondria. Ragged muscle fibers. Lab studies here, the very complex laboratory screen. This is the serum acyl carnitine. These are the labs of all the myopathies: serum acyl carnitine, low free carnitine, and elevated acyl to free carnitine, suggestive of disrupted fatty acid oxidation. Now we return to the treatment of mitochondrial myopathy. No curative treatment. Some low evidence studies suggest that coenzyme Q10 or creatine may be beneficial. These are some photos of MERF syndrome. They have myoclonic epilepsy, ragged red fibers, and this the MELAS syndrome. It refers to mitochondrial encephalopathy, lactic acidosis, and stroke-like episodes. This is the end of all myopathies, is rhabdomyolysis, breakdown of skeletal muscle tissue, membrane disruption, efflux of ions, proteins, hemoglobin, CK, seen in acute episode of many metabolic myopathies, may lead to systemic complications. This is the summary of the differentiation between glycogen storage disease and lipid storage disease and mitochondrial myopathies. I think this is the most simplified one. The next coming we will not go through, but this is the most simplified one about the symptoms, and then the most diagnostic test as regard the blood sampling, resting and post-exercise or progressive exercise test, and the result of muscle biopsy. And lastly, the treatment. Here the resting blood samples in glycogen storage disease elevated CK, in myocardial disease elevated uric acid, in lipid storage disease elevated acylcarnitine, in mitochondrial myopathy elevated lactate and pyruvate. About the exercise test, the post-exercise blood samples. In glycogen storage disease, low lactate. In lipid storage disease, possible elevation of acylcarnitine. In mitochondrial myopathy, elevation of lactate and pyruvate, and characteristic minimally change in venous oxygen concentration. About the muscle biopsy, the most characteristic is ragged fibers in mitochondrial myopathy. You may see glycogen content increased in glycogen storage disease. You may encounter elevated lipids in the muscles in lipid storage disease. About the treatment, it relies on two corners, the carbohydrate diet and the lipid diet, and the exercise adaptation. Glycogen storage disease, carbohydrate supplementation, frequent low dose carbohydrate supplementation with glycogen synthesis or breakdown conditions, low dose carbohydrate, restricted carbohydrate diet with glycolysis conditions. As regarded the lipid storage, we stress on low fat diet and high carbohydrate diet, low fat diet, high carbohydrate diet. About the mitochondrial myopathy, carefully regulated exercise training, avoid fasting, maybe creatine or coenzyme Q supplementation. And the rest of the presentation we shall not discuss, only for reference if one wants to return to it. And thank you all.
Thank you very much, my dear Professor Dr. Hanan, for this very deep and very illustrative lecture. I have, do you listen to me, Dr. Hanan?
Yes.
Yeah.
Yes,
for this
Professor.
For this very nice and deep presentation. And just I have some key points about this lecture. First one, about anaerobic oxidation. Anaerobic oxidation is needed when there is vigorous exercises, because it is faster than aerobic oxidation and the citric acid site formation, citric acid cycle formation. It is needed because it is 100 times faster than the fat oxidation and the normal glucose oxidation. For this, in vigorous exercises, anaerobic oxidation will work a bit. In the result is lactic acid. But in case of failure of this anaerobic oxidation, lactic acid will not be elevated, as Professor Hanan told us. Anaerobic glycolysis, we have glycogen first. Glycogen reside in either cytoplasm or in lysosomes. In cytoplasm, glycogen should be degraded to glucose to enter the anaerobic oxidation. But if there is a defect in the glycogen, there will not be glucose formation or lactic acid formation or the 2 ATP energy high
دكتورة حنان، بس من فضلك تعملي ميوت او يعني تعملين الهوست بقى بعد اذنك يعني.
دكتورة حنان،
انا عامله ميوت، بس عامله ميوت يا دكتوره
على اسمي بقى، معلش كليك يمين على اسم انا وميك مي ذا هوست يعني
في البارتيسيبانتس يعني
على اسم انا بس، اسم انا في البارتيسيبانتس.
حاضر. دكتور عماد.
ايوه.
مور
ايوه.
ميك هوست.
ايوه. تمام كده.
تمام
ماشي، شكرا.
Then
mute again.
we have in glycogen metabolism, we have either a defect in the glycogen or a defect in the glucose metabolism. In glycogen, we can see a defect in either myophosphorilase, which disrupt the bond. عماد الصوت مش
انا
واضح خالص.
هعمله. خلاص لحظه واحده معلش، في حد بيتكلم.
مفيش فايده.
لا لحظه واحده معلش.
Eğer bu denizlerde bir gün göçü kopacak olursa, yangın yerine dönecek ilk liman iskelleri.
او خلاص تمام كده خلاص.
Then we have a defect in the glycogen metabolism in which there is no debranching, no dissociation of the bonds, the linear bond 1,4 bond of monosaccharides or the 1,6 bond vertical one. Then the linear enzyme which disrupt the linear monosaccharides from each other is known as myophosphorilase. And the other 1,6, which is vertical, is known as the branching or the brancher enzyme. If there is a defect in either myophosphorilate or the brancher enzyme, this means that the glycogen will not be transformed to glucose. And for this, the patient will suffer from weakness, cramps, and myoglobinuria and rhabdomyolysis. And as Professor Hanan told us, when there is exercises after about 10 minutes or quarter an hour, the normal aerobic oxidation either by glucose or by fat will restore the energy for the patient and will go on his exercises. But if this not occur, there is a defect also in fat metabolism, which is theoretic. Now, restoring the energy after 10 minutes or 15 minutes, this means a second wind. Second wind means that the exercise will be improved after 10 or 15 minutes. This is known as second wind. Here also there is another phenomenon which is out of wind phenomenon. Since the defect in glycogen, not in glucose, when we ingest glucose as sucrose or disaccharide or monosaccharide, will enter instead of the glucose, which is blocked from coming from glycogen. Then there is improvement in the exercise. This is known as non-out of wind, because if the defect is in glucose metabolism, glucose will not improve the problem. Then we have in glycogen defects, we have second wind, we have improvement with using glucose or any disaccharide or monosaccharide. There is improvement in the exercise, and the end result, there is a defect. If there is a defect in glycogen storage, we can
في سؤال يا دكتور علي معلش. السكند ويند
اخلص بس اخلص يا اتفضل دكتور.
لا السكند ويند انا اقصد ان هو يعني مثلا حالات الكوليديكيشن بين نيوروجينيك بيستريح لما بيقعد ويحرك تاني بيتحرك، فايه ايه الباثوجنومونيك؟
No. In this, glucose will enter in the anaerobic oxidation instead of the glucose which is coming from glycogen. In second wind, this means that after 10 to 15 minutes, the fat metabolism will work. The fat metabolism will work, not and the aerobic oxidation with glucose also will work. Then the defect will abolish, because we are not now in need of anaerobic oxidation. After 10 to 15 minutes, the fat will work, and the working of the fat means that the defect is not present.